So some things in the chat here. Andrew would like to know what my favorite microorganism to study is. Wow, that's a good question, Andrew. Where do I start? Hmm. Um, well, it has changed over my years of experience with microbes. Currently, my favorite one to study is HIV. And down in the research lab on the first floor, we're starting up uh, slowly over the past couple of years. I've been starting up my research lab with the goal of studying how HIV gets into the nucleus of your infected cell as and kind of the idea of how it parasitizes your cells and kind of takes over your secretory system and your cell signaling pathways and is really good at evading the immune response because, for instance, an HIV virus particle looks a lot like self in the envelope in the lipid bilayer of this HIV particle. For instance, you find things like CD4 and MHC1 and things that normally are on immune cells. It pops it right into its virus membrane, and it's a really sophisticated virus and how it parasitizes your cells. So that is my current favorite. There's been times where phages had my attention. There's been times when hepatitis C virus during my PhD, I was studying that. So always viruses for me, but the target has shifted over time. And I love that because viruses are packets of genes doing what genes do. And you guys know what genes do. What do genes do? There's two main things genes do. And if you can understand that, you can understand what a virus is. So what are the two things a gene does? Genes replicate, very nice. That's number one. And they make protein. And of course, protein goes from gene to mRNA to protein, right? So that's what viruses do. They take over your cell, they take what they need, and they make copies of themselves, and they make mRNA transcripts and proteins. And once they make everything they need, the virus self-assembles, spreads on to the next cell. There you go. So viruses are applied cell and molecular biology. Very nice. All right, so thank you for that question. Andrew tried to get me off on a tangent before class even had a chance to start, right? That's like asking Dr. Jones anything about T cells. And then he goes on like a 20-minute rant about T cell biology, right? So for me, it's viruses. Okay, nice to see you all. Thank you for logging in. Thanks for the cameras on. I do appreciate it. It helps me from having COVID flashbacks from when I was locked in my basement for 2 years teaching on Zoom all the time. So it's nice to see smiling faces and I appreciate the effort to have cameras on and participate if things are appropriate where you are, right? We don't want to see inappropriate things in the background. So again, I do appreciate it. All right, so any questions for me before we get started? I'll pop our slides up here. Any questions? That's my PowerPoint slides. I am on my laptop, so you can pop things in the chat, but it will be easier. If you'd like to answer, raise your virtual hand, just unmute and ask questions. It's okay. This is supplemental. This is casual. I'm wearing my polo shirt today, right? It's casual Wednesday instead of casual Friday. Spring break is in the air. So please, this can be interactive. I don't mind interruptions, etc. All right, so no questions? A little more practice with the gram positives. You ready? Gram positives practice. All right, there we go. Because this is Zoom, I think I can send you a poll. So let's do polls on these questions. And there you go. I see about half of us have answered our poll question. I will give you another 30 seconds or so. Here we go. And this is why I like polling. I do miss, right? Need to practice my poll everywhere skills and find a happy medium with what we do with poll everywhere because look, this is a wonderful poll. There you go. There's your results. And why I think it's wonderful is it means I can teach you something, right? I know where the deficiencies are and we can target it, right? So that's wonderful. We have pretty much we have pretty much evenly chosen A through D, except for choice E, poor choice E. What's going on with choice E? Why did nobody pick E? That's a very special auger, right? That's one of those ones like one auger, one thing. What do you, okay, Mrs. Abraham, what's up?
Hello, Dr. Manna. I thought for E, that's only for like Mycobacterium species. So he,
Correct.
to me, doesn't look like he has that. So,
Absolutely. Very nice. That's one of those special ones, like one auger, one genus, Mycobacterium. Um, blood auger is not like that. Right? Blood auger is like everything grows on it, helps you narrow things down. Some augers are very specialized, so very nice. That's why we memorize these things because you pair that with the knowledge of what mycobacterium infections are, right? You can say, hey, this is a little kiddo who's not coughing up blood and has respiratory TB, doesn't have leprosy and skin lesions, right? That's how you cross things off. All right, so let's see. We've got pharyngitis, a severe case of pharyngitis. So much so that the redness and irritation you normally see has coalesced into a fibrous, adherent membrane on the back of the throat, right? We can see that. Let's see here, right? What? Right there, right? That's normally exudate, what you're seeing there, except for, right, normally you would be able to scrape that off, take a sample, that would be pus and inflammation, little white patches. This is adherent, right? And now, one important thing in the question. Sorry, I'm doing this without an old-fashioned mouse. I'm using my trackpad to draw. And sorry, I have a question in the chat. This is the supplemental PowerPoint. Yes. Did I forget to post it on the Canvas page? Okay. So I did. Fair enough. Do you all, well, I'll just ask you, do you all want the PowerPoint slides? Would it help you with note taking, etc.? Is it worth me taking a minute to pop it on Canvas? Yes. Okay. All right. Thank you for letting me know. So let's see. How do I do this? Stop sharing. Right here I go. I thought I had everything all set up. Canvas. All right. Bear with me. Look for the right dog picture in my Canvas dashboard. Have you guys favorited my course? Put it on the dashboard. You see the dog pictures? That's what we do in Micro and Immuno. We have dog pictures. I never remember your course name or your course number, but I remember which dog picture is your course. The BSCN 600, something like that. Right? That's not as fun as cute dogs. All right, here we go. All right, through the magic of the internet, this should be up on your Canvas page in a few seconds. In the week 13 content page, all right. You're welcome. All right, and oh, good. We got some discussion in the chat too. All right. white patch looks like strep, but it's not quite. Oh, that's not one thing. I like the way our chat is trending here because, oh, and now you can see the answer, right? Okay, we'll just skip that. If you're looking at the bottom, you can see the answer. All right, a little peek behind the curtain. But pharyngitis, right? You have a sore throat. I want you to think strep, right? Strep throat. What causes strep throat? Which strep? There's a lot of streps. There's two names for it. You can call it a group something, or you can call it by its real name. What strep causes strep throat? It is group A strep. Very nice, Aya. And now somebody follow up with the genus and species names. It is strep. Group A strep is strep. Strep what? Is it strep agalactae? Is it strep pneumo? Strep pyogenes. Very nice, very nice. It's strep pyogenes. All right, so what's really key in this question is one little keyword. Again, micro, a lot of it is finding the keywords in your question. I'm going to underline it again, my wonky line. And right there is the keyword. Vaccine history is unknown. Okay. Whenever a micro question tells you patient history and talks about vaccine status, you're at a major decision point for your differential. Okay. And what I mean is when we have all of our learning objectives for the pathogens, that includes diagnosis, treatment options, right? Epidemiology and transmission. All these tie in. Basically, you need to memorize which bugs have a vaccine and which don't, right? Because many bacteria can cause cause a sore throat. Strep, group A strep throat. You could have diphtheria. Somebody mentioned diphtheria in the chat. You could have a sore throat from Haemophilus, which we'll cover in our next class session. Plenty of bacteria can do it. Not every bacteria has a vaccine in this scenario, right? So you have a sore throat and you have a patient that is non-vaccinated, and the ones we looked at today that cause sore throat are diphtheria and group A strep. Which is it, right? Which one has a vaccine, which one doesn't? That's the key to answering this question correctly. So, which one has a vaccine? Diphtheria or group A strep?
Diphtheria?
Diphtheria. Very nice. Bonus points for knowing what kind of vaccine it is. Do we remember what kind of vaccine it is? You find it in DTaP. Let's see, it's a toxoid. Very nice. You take that toxin, you modify it so it loses its pathogenesis, and then you inject it into the patient so you make IgG to the toxin, right? And that's because diphtheria toxin is really dangerous if it hits your bloodstream. It can cause myocarditis. It can affect the way your heart muscle cells work. So that's why you want to make an antibody to the toxin and neutralize it before it hits your heart. All right, so that's the basis of diphtheria toxin. Now, Amir had a great question. What is the white plaque called again? Now, it would be starting as exudate, but diphtheria has a special name here. What we call this, you call that the pseudomembrane, because that is not plain old-fashioned exudate there. That is not scrapable. You cannot sample it and take a little sample and do a culture. If you try to get a sample of that, it will all come off in one big piece and it will start bleeding. It's a fibrous tissue, adherent. Diphtheria has nasty toxins. It kills cells. And so you call that a pseudomembrane. That is different than what strep throat exudate will look like. Little white patches of like pus. Good question there. All right. And mentioning the toxin, I see something popping up in there, is combined. Yeah, Tdap is tetanus, diphtheria, and acellular pertussis, 3 different toxoid vaccines. All right. So yes, that is a good way to differentiate it. Now, speaking of the toxin, you got a mechanism for diphtheria toxin. How does diphtheria cause a pseudomembrane, hurt your cells? How does diphtheria toxin cause myocarditis if the toxin hits the blood and goes into your heart? We do need to memorize toxin mechanisms. I will test on them. I promise you, I will test on them. You don't need, not just like it's called diphtheria toxin A or whatever it's called. I don't, right? It's what's more important is you know the mechanism. So diphtheria toxin, how does it work? It's a phage-mediated toxin. It's mediated by bacteriophage transduction. So actually, diphtheria cells need to be infected with a virus to get this toxin gene. Really cool. All right, how's the toxin work? Go ahead.
I have a question about how the toxin works. Is it similar to like candidiasis where it sits on the heart valves and that's how it affects them, like it gets in the bloodstream and sits on the heart valves?
Ah, what you're describing is
Hmm.
called endocarditis, where an endothelial cell or a heart valve becomes colonized with a pathogen. It can be
Hmm.
fungal, like Candida. More
Hmm.
often, it's actually a bacterial pathogen like Staph aureus or
Hmm.
strep species because they have big
Okay.
sticky capsules, right? And make biofilms. But Candida also can do this. What diphtheria does, diphtheria is different. The bacteria is not in the blood, the
Hmm.
toxin's in the blood. And the
Okay.
toxin is what's toxic to the heart and causes the myocarditis symptoms, right? Because myocarditis, just by the term, means heart inflammation, heart muscle inflammation.
Mhm.
And it can be because there's like a virus infecting your heart cells, an
Mhm.
intracellular bacteria, or a
Okay.
toxin that's causing inflammation.
Okay, because I remember seeing that also like with candidiasis and that they have the white plaques in the mouth, the children and stuff. So that's why I was a little confused
Hmm.
about it. Okay.
Sure.
Thank you.
Those, again, are not adherent to, you can scrape those off.
Yeah, that was my question. Like, if it didn't say vaccine history, if it was scrapable or not, could that help as well in understanding this?
Absolutely. Absolutely. All right. Sadaf.
Dr. Man, I can explain the diphtheria toxin that you asked.
Okay, please do. How does it
Yeah.
work?
So we have a B subunit and an A subunit. The B subunit binds to the host cell receptor and the toxin gets inside, so through the endocytosis, and then you have A subunit which escapes into the cytoplasm. And the A subunit has like a ADP ribocyclase EF2, which elongates into factor 2, and that's how the diphtheria toxin works.
All right. Very nicely described molecular mechanism. That's it. It blocks EF2. I agree. With Andrea and Jasmine. That's very nicely done. And that's the level of detail we want, right? Like we don't Sadaf just went like a little over and above the needs, which is fine, right? Because that's like knowing these things is the context, right? Like tagging a Y to a factoid, right? Because A B subunits, we know what those are. A is activity, B is binding, and it's got to bind to a cell and be delivered and all that stuff, right? So for our course, right, diphtheria toxin, it has a name. I believe it's called the AB diphtheria AB toxin, whatever its name is in the slides. Make sure you nail that. And then it blocks protein synthesis by blocking EF2, right? We want its target and what the outcome is, right? So we have a few bacteria toxins that target our ribosomes, right? Some block elongation factor 2. You know what that is, right? From genetics and biochem. Some go after the ribosomal RNA subunits and cleave them and then activate the ribosome that way, right? So we need to know it's a protein blocker and how it happens. Or this bacteria increases cyclic AMP, right? We had a anthrax toxin that targeted cyclic AMP. And what that happens is it moves ions in a cell and you get edema because you lose fluid in the cells, right? So again, we need mechanisms here. All right, now, can we clarify where vaccine history is key? All right, yes, Amir, I absolutely can. And this is why you need to, when we have a bullet point in our micro slides that this pathogen has a vaccine, you take note of that and you memorize it. Because in this week's content, right, we have like a narrower focus. We literally studied two things that could cause a sore throat, right, Amir? And one is strep throat and one is diphtheria, right? So we have a vaccine for one and no vaccine for group A strep. So when we mention vaccine status in a question, it's for good reason. It's for you to think of which pathogens have a vaccine, and whatever we tell you in the question, you go the other way, right? So unknown vaccine status history, right? All that, that's just our way of saying patient's unvaccinated, right? And if we say UTD, right, because again, this is biology, we abbreviate everything, up to date on vaccines, you just take for granted that they have all their vaccines, and then you forget about diphtheria in that scenario, right? Because think of it this way, don't go into a question assuming that like, hey, this is the 1% of the population that doesn't respond to the vaccine, right? It's not a trick question. We're giving that to you as a really good little nugget, like a clinical nugget to make your decision-making process easier. Okay, so whenever a micro question mentions vaccines, please take note. All right. Good questions, guys. All right, so let's finish up this this question here. We had some really great discussion. Glad we're thinking. Now, because it's diphtheria, I'm going to relaunch the poll. Because we had a good discussion, I think we narrowed in on the choice. Let's leave the new poll open for like 15 seconds and see how we confirm it could be diphtheria. Right, and there you have it. Now we're honing in on the right answer, which is the ELEC test. The ELEC test is a toxin-based test to look for diphtheria. Catalase test is, excuse me, to differentiate who? What do we use the catalase test for? It's like the oldest micro test in there, Staph and Strep, right? Staph and Strep, very nice. Okay, so let's see. Whoop. Where am I going here? Okay, so this is what the ELEC test is. It's literally just a test for the toxin. Okay, it looks for a precipitation of the toxin. And how they do that is they put an antibody in the dish, a monoclonal antibody that binds to the toxin. And when the antibody and the toxin come together, they precipitate, and it makes a little white line in the agar dish. No big deal. But the key is it's a toxin test. Why do we have to test for the toxin and not do like a culture test or a PCR test or serology? Why can't we do those? Yeah, that's what's pathogenic, right? I saw it in the chat. I think Sadaf had her hand raised, sorry, but it's that's it. It's like you have normal flora called diphtherioids. They're Corynebacterium genus bacteria. They are normal flora in your nasopharynx and they don't hurt you, right? Until it gets infected by this bacteriophage that gives it a toxin, and then it becomes pathogenic. So you do a culture test, you do a PCR test, whatever other test that just sees if the bacteria is there, it's going to be there, and it's always going to be there. You need to see if it's the dangerous type, right? So you're looking for what's pathogenic. All right, next question. Next question. Try to pick up the pace a little bit for our remaining 30 minutes. All righty, let's check our poll. This is bringing a little immunology with you, but to really key in on the question here. We need to understand the microbe, what it's doing. All right, now here's another trend I want you to be aware of in microbiology questions. Right, because there's a lot of terms we use commonly, right? Patient's up to date on vaccines, patient has incomplete vaccines history, right? When we say this, my favorite virus, what is that a way of saying? Patient has a history of HIV infection. Why do you care? Why do you care? Because the patient is what? I see. Go ahead. I see a hand raised. It's in the chat. It's immunocompromised. Very nice, guys. Very nice. So that's just another way to get you thinking, make clinical associations, right? Because it's a little too easy to say you have an immunocompromised patient and here's their symptoms, right? So we just make it a little trickier by saying these types of things. All right, so we have trouble breathing, night sweats, and bloody sputum. And then I see a histology slide, path slide, that looks like a circle. So you have a bloody cough, somebody who has a fever that gets higher throughout the day. What's our cause, right? What's the etiology here? This is TB. I see it in the chat. And we're talking about Th1 stuff. Andrew's already beating me to my follow-up question. All right. And it's a Th1 response. It's an intracellular bacteria, right? Macrophage gobbles it up, tries to kill it. It doesn't succeed. When things become persistently infected and your immune system can't close it off and kill it, what do? Make a granuloma. Put a circle around it. Try to keep that pathogen in place instead of spreading. That's exactly what happens. All right, so if this is a Th1 pathogen, let's see how our poll's doing. Very nice. If this is a Th1 pathogen, endogenous interferon gamma. Excellent. That's the correct answer. Basically, give Th1 pathways a little nudge, right? In theory, is a way you could help the patient out, right? Because penicillin's not your drug of choice. What's going to happen if you give them IL-10? What's going to happen if you give them IL-10? IL-10's an immunosuppressant, right? Yeah, that's not good, right? That's not good. You do immunosuppression, you're going to have the granuloma rupture and the bacteria is going to escape, right? Have you seen all the new commercials for all these new monoclonal antibody drugs? Let's see, one's called Humira for like rheumatoid arthritis. You have inflammation of your joints caused by the immune system, and they prescribe a monoclonal antibody that blocks TNF alpha. Okay. And at the end of that commercial, what do they say? They say, let your provider know if you have a history of TB infection. Because if you have a history of TB infection and you take a TNF blocker, guess what happens to your granulomas? Right? They open up, they release the bacteria. You're losing that immune. How would sorry, and the question, how would a ruptured granuloma present? It would present, great question. It would present by miliary TB. Miliary TB means the bacteria's got out of that initial granuloma, hits the body, bloodstream lymphatics, and it spreads to other sites. Where exactly it goes, I can't tell you. Different from patient to patient. Bones, joints, CNS, tissues. It goes all over the place. So that is what you would see. All right, very nice. Next question. All right. Do a little toxin practice, supplemental, nice, easy. This is just a recall question. All right, so sulfatides. Sulfatides, poll's looking very nice. What is the function of sulfatides? Let's see. Is it choice? All right, so, all right, well, I see a hand. Sadaf, do you want to answer?
Yeah, I would love to explain the function.
Okay,
So
go ahead.
sulfatides, they block the phagolysosome fusion. So basically, in that situation, the macrophages eat the TB, but they cannot destroy it, and the TB still survives inside the cell.
Very nice. Sulfatides are the reason why macrophages can engulf TB and not kill it, right? Blocks the lysosome from fusing with the endosome, right? When we say intracellular bacteria, got a got an insider secret for you, right? I got a tip for you. When we see intracellular bacteria, most of the time they're inside a macrophage or a neutrophil because they target those cells for infection because they know their job is to endocytose bacteria. Very few intracellular bacteria go after like an epithelial cell, an endothelial cell. They're almost always inside of phagocytes because they don't care. They want to be engulfed. That's where they live, right? So what don't they have? They don't have capsules. They don't, right? They don't worry about blocking phagocytosis opsonization. They just go for it and then get where they want to be and then they resist killing. So very nice. All right, now let's see. Of course, we're going to get more practice with our question. Adhesion to respiratory mucosa. Name a virulence factor that can help a bacteria adhere to your respiratory mucosa. It's a similar structure, and I think we talked a little bit about this in week 1. They're very similar structures. They have slightly different shapes. We call them 2 different things. They're both for adherence. Let's see what we have in the chat here. I see pili. Okay. Fatima, that's really close. That's really close, right? So, and I think if we talked about this before, I actually was looking this up last night to make sure I had it down too, because we have these things called pili and we have these things called fimbriae. Okay. They're made of the same subunits, the pilus protein, and what differentiates them is 2 things, their length and the protein they put on the tip. Okay. And here's how it works. And make sure I'll clarify this for you all. Is a pilus is always for conjugation and sharing plasmids, right? So the pilus is a bigger, longer, thicker straw-shaped structure, and the tip protein is designed to bind to another bacteria, shoot a plasmid across. Then we have fimbriae, which are shorter, more hair-like structures, not as thick, and the tip of their pilus gene is to attach to mucosa, right? So fimbriae are the shorter hairs that are made of pilus subunits that are for mucosal attachment. A pilus is always for conjugation and plasmid transfer, right? Same proteins, different outcomes. So for this one, adhesion to respiratory mucosa, best answer is fimbriae, remembering that fimbriae are made out of pilus proteins. Okay, hopefully that helped clarify. Sadaf, do you have a question?
No, you ask if we know an example. I just wanted to answer that.
Okay, gotcha. Well, I
So
beat you to it.
do I say it?
Sure.
So the one example is the beautiful E. coli UTI. They have like P. fimbriae pili and it's stick to uropithelium. And the next one is Neisseria. I believe the gonorrhea, I cannot say it very well, type. And they use pili as well. They stick to the mucosal cells.
Mhm. There you go. You mentioned 2 very common mucosal pathogens. E. coli has a fimbriae to attach your urethra and cause a UTI. Gonorrhea has a pilus to attach to GU mucosa and cause an STI. Absolutely. Those are real life examples of fimbriae. Very nice. All right. In the super cool. I agree. That is super cool. That is like the biological basis of disease right there. Block perforin granzyme mediated killing. Do bacteria do that? Yes or no? I'm seeing head shake. No. Perforin granzyme, who kills that way? We have, go ahead, Mrs. Abraham.
Oh, NK cells do that in cytotoxic T cells, and it's
Yeah.
mainly for viruses, not
Yeah, you got it. So bacteria don't really care about that. Absolutely. Cleave host cell RNAs, we'll see those next week. That is one way that some gram-negative bacteria shut down your protein synthesis. They cleave your ribosomal RNAs. Disrupt chemotaxis by neutrophils. That's another TB virulence factor. What do we call that one? Which one blocks neutrophils from coming to the scene? We call that one C5a peptidase, not quite, but that would be involved. I think C5a peptidase is in group A strep, if I recall correctly. But that would help block chemotaxis. You're absolutely right. Right mechanism, wrong bug. For tuberculosis, it's a different name. I believe we call it the cord factor. I believe it's called the cord factor. That blocks neutrophils. And why that happens, or the way I memorize that, is TB wants to be inside a macrophage that needs a little Th1 nudge from a helper T cell, not a neutrophil, which is like an innate killer. And neutrophils don't really need that nudge to be a good killer. All right, so they don't want to be in neutrophils. They want to be in macrophages. All right. Next question. Right. Exploring post-streptococcal hypersensitivity reactions in this question. There are a few pathogens that are exam question writers' favorites. Tuberculosis is one, because that's a great way to ask you about DTH reactions, type 4 hypersensitivities, Th1 pathways, or a micro question about TB. And then we have strep, group A strep, because we can ask questions about the micro, or we can ask questions about the post-infectious hypersensitivity reactions that can occur and hurt the patient. There's even a toxin-based complication for group A strep called scarlet fever. That's not a hypersensitivity, but that's by the toxin hitting the bloodstream. One of the super antigens, by the way, strep pyogenes has super antigens. But this one is exploring post-streptococcal GN glomerulonephritis. And here's how post-strep complications happen. We have two of them. All right, first and most common is rheumatic fever, where we have a molecular mimicry reaction. A protein on the surface of group A strep looks a lot like myosin, and you make antibodies to this group A strep protein. And once the bacteria is killed off, the antibodies have nothing to do, and they're floating around your blood, so they wind up binding to your heart. And you have antibody binding directly to tissue, causing inflammation. What kind of hypersensitivity reaction is rheumatic fever mainly? If we have antibody binding to a strep protein, then to your heart, very nice type 2 hypersensitivity. And the way this works is you have strep throat that can lead to rheumatic fever. Strep throat usually, almost never, does not lead to kidney problems. Now, when you have impetigo, skin infections caused by group A strep, same microbe. Oh, sorry, let me back up one second. What is that magical protein in group A strep that the antibodies are binding to for molecular mimicry that wind up binding to your heart and causing rheumatic fever? We need to close up that loop there. That is M protein, very nice, a surface protein. That is for anti-phagocytic, protection from immune system, as well as attachment to your mucosa. Very nice. Now, when you have skin infections with group A strep, you do not get rheumatic fever after that. You get a hypersensitivity reaction where antibodies bind to strep bacteria in your blood, and they start to precipitate in areas of your body where the vasculature is leaky. And for group A strep, it happens in the kidneys, right? Post-strep GN. And similar types of reactions to this, where antibodies might bind a pathogen and precipitate out in the leaky parts of your vasculature. You also can see those in the skin, right? In your small capillaries, and you have rashes, or your joints, right? Because your joints have leaky vasculature as well. What kind of hypersensitivity reaction is that? When you have complexes settle out and precipitate and cause inflammation. I'm seeing a couple threes on people's hands. I'm seeing 3 in the chat. That is a type 3 hypersensitivity reaction. Very nice. So post-strep GN, we can cross off type 1 and type 2 hypersensitivity. And our pollen, right? Because some of us went with type 2, choice D. And we cleaned up why it's not type 2, right? Because type 2 is for rheumatic fever. So if you have antibodies binding to bacteria, they precipitate in your kidneys and you activate complement. What kind of complement? Is it classical or alternative? Which one uses antibody? And of course, it is the classical pathway. That's your correct answer. Classical pathway, right? And that's just a tougher way of saying, is it type 2 or type 3? Right? Or right, because alternative, that's your baseline, that's innate, that's kind of like always going in the background. Next question. Oh, I'm sorry, I forgot to launch your poll. Here you go. I will not open it. I will not keep it open very long since you've had plenty of time to look at the question. Okay, and this is a great example of a tough exam question. Tough and fair. You're not going to have all questions like this in your test, but I think this is a great example of where you're headed, where I want you to be prepped for the exam. All right, again, we'll do some first order recall. We'll have some questions where patients present with symptoms and we tell you diagnostic tests and you tell me the name of the pathogen. And then we're going to have some like this where you diagnose in your head and then you bring back out a factoid, a second order jump. So what we have to do is first find out what is the organism, right? And there's a couple ways to do it. I saw in the chat I was asking about staph epidermidis. And I like your thinking, Aya, because there's a couple things. One is the memorization and the factoids, right? What do we memorize about staph epidermidis? Did you memorize that it's novobiocin sensitive and catalase positive and coagulase negative? Those 3 factoids together tells you it's staph epidermidis, right? And then, right, because these things you want to, and it's a prosthetic valve, and that's where I was heading next. Very nice, is also you take those things that you memorize, and you can cross-check it, right? You can verify, right? Don't just make that association like I see these words and I know it's absolutely staph epidermidis. Take a moment to double check. And what I mean is it's a prosthetic valve infection of the heart. And when you have staph, right, You have Staph aureus, which does everything, including binding to native valves. It doesn't bind to prosthetics, it binds to native valves. You have Staph saprophyticus, which causes UTIs. And you have Staph epidermidis, which binds to catheters, binds to prosthetics, and gives infections in the immunocompromised. Right? So that the infection plus the biologics that she had memorized makes it a slam dunk. It is Staph epidermidis. So then it's non-hemolytic. Very nice, Drew. It is non-hemolytic. Your correct answer is gamma. Doesn't have hemolysis, right? So a good example, and these are the buzzword things like know what the hemolysis patterns mean, right? Be able to look at a blood agar dish and see zones of clearing or no hemolysis, know it's alpha, beta, gamma, and know what the description of them is too, right? That is very helpful. But this is gamma hemolytic. All right, the other staphs. Let's practice our staph hemolysis patterns now. Staph aureus. What is Staph aureus? All right, I is definitely watch Sketchy then because beta would be aureus shining gold. All right, is that the one with like the golden staff or something like that? I watched that years ago. I still remember some golden staff that's supposed to be beta hemolytic, right? See, Sketchy works, right? These associations, these Y's. All right, so yes, Staph aureus is beta hemolytic. What about Staph saprophyticus? That is gamma, same as epi. So how do you tell apart, other than hemolysis pattern, how do you tell apart Staph aureus from the other two staphs? What do we call the other two staphs? There's a clinical term, right? In the future, as clinicians, you're never going to say Staph saprophyticus. You're never going to say Staph epidermidis. You're going to say coagulase negative staphs because that's a lab test that lets you know it's not Staph aureus, right? That's like physician code for saying, oh yeah, it's a staph infection, but it's not the serious one. Coagulase negative, right? Because Staph aureus has coagulase, and the other two do not. All right. 946. Oh, look at, and I love this picture. Right there you go. Alpha, beta, gamma. The picture says 1,000 words. An artistic grad student took 3 different bacterial cultures and wrote Greek letters. All right, last question. Last question. As I understand or I hear that you guys have genetics or biochem review after this. We had questions about alpha hemolysis in the chat. Yes, no staphs are alpha hemolytic, only streps. Most notably, the viridins group strep, named viridin because the Latin word for green is viridi. There you go. I taught you some Latin today. And alpha hemolytic patterns look kind of greenish brown. So alpha hemolysis is the viridins group strep, the strep that hangs out in your mouth, and strep pneumo is alpha hemolytic as well. Strep pneumo is a viridins group strep member. There you go. Look at that. We're talking linguistics now too, right? And again, it's these ways, these things I'm telling you, these little like stories that I associate with memorization points, that's the same thing Sketchy does. You're tagging a Y to a memorization point. Don't just memorize strep pneumo is alpha hemolytic in a vacuum. Give it some Y. Doesn't matter if it's a silly cartoon or an actual mechanism. A little context goes a long way to recall. Right. Now, there are 2 ways to infect a fetus. Pathogen either hits the blood during gestation, crosses the placenta, and infects the baby. That's usually when mom doesn't have IgG to cross the placenta and protect the baby, usually a primary infection during pregnancy. Or during delivery, the baby can encounter bacteria, viruses that are in the vaginal canal. You're right. During delivery, things that might be vaginal mucosal bacteria or fecal bacteria, GI bacteria, right? So things like that on delivery, you want to watch out for things like herpes simplex virus. You want to watch out for chlamydia and gonorrhea, You want to watch out for E. coli. You want to watch out for group B strep, because during delivery, a baby can be colonized by group B strep. And group B strep is not such a big deal to an adult with a fully formed immune system, but group B strep is really dangerous for a neonate that has an underdeveloped immune response, is not great at cleaning out encapsulated bacteria because it has a big, tough capsule. B cell response is underdeveloped, all that kind of stuff. So group B strep hits the baby's bloodstream, spreads all over the place, and causes meningitis and hepatitis. It goes everywhere. It is a nasty pathogen for a newborn. All right. So, and this is missed, and this is right. About 1 out of 3 women are colonized by group B strep. No big deal, right? It's not going to affect your life until you're pregnant. And what you do is at some point during gestation, 35 weeks, 37 weeks, I don't remember the number. It's been too long since my wife was pregnant. My kiddo's 4 now. You do a urine culture, right? You find out if they're colonized with group B strep. And if they are, it's no big deal. You give them penicillin. Group B strep goes away and it's no danger to the baby, right? So this is like healthcare access, underserved populations. We're talking about really easy preventable things that lapses in care can manifest, right? So that's like how you do it. You do a urine culture at this point, right? So this is just like a micro question now asking about another diagnostic test for group B strep. And you are correct that the correct answer is the Camp reaction. The Camp reaction. That is your correct answer. That is a biochemical test to see if you have group B strep. It's a hemolysis-based test, right? You don't need to, it's where group B strep beta hemolysis meets Staph aureus beta hemolysis, and they merge and make a super hemolysis pattern, right? That's what it is. Okay, so it's a hemolysis-based test. Is PYR for group A? Yes, Delroy, it is. That's a wonderful question. Just like CAMP is for group B strep, PYR test is the new biochemical test for strep pyogenes. Absolutely. And catalase, we know what catalase is. Yep, and there you go. Sadaf says we have a arrowhead shape on the agar. That is your super hemolysis reaction. All right, Quellung reaction. There's one more I see I want to cover. An old-fashioned test for a strep species that I still see in textbooks. Clinicians tell me it's never in use anymore, but it's still in textbooks, so you got to memorize this word. If you see Quellung test, I want you to think strep pneumo. Quellung is for strep pneumo. There are times, unfortunately, I have to teach you historical diagnostic tests because, again, things lag from the clinic to the textbook, and you might see that word on a standardized exam one day. All right. I went over time. It's 9:52. All right. So questions for me? Questions for me? All right. Well, if you're one that's going to do a little work over break and stuff and you have questions, please email me. We have a few days of work. We have a few days off throughout break. I will be responding to emails sporadically when I'm working. So don't hesitate to reach out if you need help. You're not on your own all break. Help is there if you need it. We'll schedule a time. All right. Otherwise, recharge your batteries a bit, get some sun, and get ready for one final push throughout the semester. Take care, everyone.